All right. The UK becomes the first country to approve genetic editing as a treatment for a couple diseases. There's a lot to unpack here. Risks, positives, of course. A lot of people will benefit from this, but there is an ethical concern. Pandora's box may have just opened. Actually, it probably opened when China began their experiments in this whole realm. But I'd like to go over that as well. So I have a few links to pull up and share with everyone. A video to demonstrate exactly what's happening inside of us when CRISPR is deployed, the genetic tool. Got to quench that curiosity. In order to do that, we got to look at all the bases. So let's dig in. I got a Nature article to start. UK first to approve CRISPR treatment for diseases. What you need to know. The landmark decision could transform the treatment of sickle cell disease and beta thalassemia, but the technology is expensive. It's kind of interesting because you can buy a CRISPR kit on Amazon for like $130. You could edit your own DNA sequences on like bacteria and whatnot. So just found that interesting. In a world first, the UK medicines regulator has approved a therapy that uses CRISPR-Cas9 gene editing tool as a treatment. The decision marks another high point for a biotechnology that has been lauded as revolutionary in the past decade since its discovery. I'm going to scroll down a little bit. There's just a couple lines I actually wanted to read from here. Where did it go? So the two trials. The trial for sickle cell disease followed 29 out of 45 participants long enough to draw interim results. Casgevy completely relieved 28 of those people of debilitating episodes of pain for at least 1 year after treatment. Researchers also tested the treatment for a severe form of beta thalassemia, which is conventionally treated with blood transfusions roughly once a month. In this trial, 54 received the treatment, of which 42 participated for long enough to provide interim results. Among those 42, 39 did not need a red blood cell transfusion for at least 1 year. That's pretty good, actually. That's really good. Problem is side effects that maybe sometimes wouldn't present themselves right away. So let's get into that. Participants involved in the ongoing trials experienced side effects including nausea, fatigue, fever, and an increased risk of infection, but no notable safety concerns were identified. The MHRA and manufacturer are monitoring the safety of technology and will release further results. One concern surrounding the approach is that CRISPR Cas9 can sometimes make unintended genetic modifications with unknown side effects. Quote, it is well known that CRISPR can result in spurious genetic modifications with unknown consequences to the treated cells, end quote. So, I'm going to play part of this video just to understand exactly what's going on when it's deployed, and then we'll get into more of the risks.
The CRISPR-Cas9 system is a tool for cutting DNA at a specifically targeted location. The technique has already revolutionized gene editing, but scientists are always looking for new possibilities. So what else can CRISPR do?
Very well done video.
Since being discovered in a bacterial immune system, CRISPR-Cas9 has been adapted into a powerful tool for genomic research. There are two components to the system, a DNA cutting protein called Cas9,
Yep.
and an RNA molecule known as the guide RNA. Bound
That's
together,
important.
they form a complex that can identify and cut specific sections of DNA.
Remember the guide RNA.
First, Cas9 has to locate and bind to a common sequence in the genome,
Locating.
PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes.
It's interesting that they say it's a great tool. I mean, it is. It genuinely is a great tool for, taking out entire sequences. But that's also the Achilles heel of it, right? So I'm not super technical with this subject. I won't pretend to, be an expert, obviously. But one risk associated with this, reading about this, there's actually a lot that can go wrong in an individual with this one tool. One of the common ones, my understanding is that when this is locating the actual part of the sequence that it's about to edit or cut out, there's a lot of the DNA that actually looks very similar. So, looks similar, right? It has very similar sequences within it. Sometimes this locates a similar part of the DNA that it wasn't actually supposed to affect. So then it cuts out that part instead of the other part, down the helix or whatever. And it completely just either swapped or completely cut out a part of the DNA that it wasn't supposed to. Now imagine that happening within a person, right? Even if it's done correctly, this is another part of the risk associated with this. Even if it's done correctly, there are a lot of these genomes that we're not exactly sure the processes they affect within our body, right? A lot of them, we're sure it affects those specific things, right? Eye color is one of them. The gene that activates HIV, I suppose. We know big processes and the gene that affects them. However, we don't know that gene that affects the color of our eye also affects our toenails, for instance. We have no idea the other things that it might affect. So by changing the eye color, 10 years later, you find out all these other processes within your body are just messed up, right? And I want to add another layer to this. Imagine I change my genes, right, to change my eye color, whatever. Or maybe I'm a newborn baby. There's cases of this in China, actually. Maybe my eye color was changed before I was born. Fast forward 25 years, I have children. They're GMO'd children. Those children are GMO'd. And because my eye color was changed, their genes are kind of messed with artificially. And we don't know the long-term effects it has on that kid. So while it seems like, the eye color has changed, everything is good, it affected me, it gets passed down, those affected genes, obviously. So, yeah. And, with that, the first Chinese edited babies, a leap of faith in science. This came out of China. So, China is one of the leaders in the genetics game since February 2014, when Chinese scientists published the results on the use of CRISPR-Cas9 technique in one-cell stage monkey embryos. They've been the main drivers of this new technique. November 2018, media from all over the world reported that 2 twin girls had been born with modified genes to make them HIV immune. Their birth was the result of an experiment conducted by He Jiankyu, did I say that right? With couples in which the males were HIV carriers. Using CRISPR technology to immunize the babies against the HIV virus, they managed to disable the CCR5 gene that enables HIV infection. However, Chinese existing regulation, thought not very detailed, does not provide legal basis for this experiment. So in particular, the 2003 ethical, blah, 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 blah. Let's skip around. Okay. So this is actually the important part. Furthermore, in spite of the alleged reason for genetic intervention related with prevention of HIV, the scientific community also knows that the CCR5 gene is related with major brain functions. Okay, so this scientist may have done some kind of human enhancement by creating two especially intelligent human beings with better memory and higher IQ. So, I mean, you know, China's doing some crazy stuff. I mean, just this week, they've been experimenting with chimeric monkeys for quite a bit. That's human monkey hybrid. This week, though, they kind of passed a threshold where they were able to get over like 50% of a certain part of the genome to be human, not monkey, if I'm remembering that correctly. But I have it right here, right? Live birth of chimeric monkey with high contribution from embryonic stem cells. So I made a video about this previously. You can go watch it. But they're kind of playing with fire. You know, they're trying to increase IQ of babies through genetic experiments. They're, you know, merging human monkey. It's getting pretty weird, you know, and the odd thing about this is that when China is conducting all these like wacky and like pretty unethical experiments, it sucks, but some countries are looking at that as like, hey, someone's got to do it. You know, someone's got to experiment on these things, because there's a lot of important data points that come from it that we're not going to do because we're moral. And, you know, I'm glad other countries aren't doing this, but unfortunately it's seen as kind of like a good thing, like a net positive that China's doing it. And just, we're reading about this stuff. Imagine the stuff that they're not telling people that they're experimenting on. It's a scary thought. I mean, when it comes from genetics, you know, these things can have such lasting impacts. It doesn't even need to be widespread, right? You get 2 or 3 successful experiments that not a lot of people are aware about. There's not a lot of research on, blah, blah, blah. You change the genes of like 10 people in China, right? And then any country, I'm not going to use them as an example. Change the genes of 10 people in one country. They have kids. What does that do 20 generations later to a group of the populace, right? Using CRISPR technology. So, just an interesting thought experiment, right? It would change a significant region, you know, in some way, and it's unpredictable too. I suppose what could offer some consolation with this is that as we develop more intensive computational capabilities, we start including artificial intelligence in a lot of these scientific models. You know, we start getting closer to quantum computing. We'll be a lot more capable of performing CRISPR treatments without a lot of the risk, because we'll be able to identify, okay, this part of the genome affects that part of the genome. You know, this process can be isolated to these 3 things. So, you know, I'm a little skeptical right now. I think it's great for the people who want to use it to benefit from on their personal accord. I'm just a little bit skeptical, big picture. So, anyways, I will leave it at that. Hope that was somewhat informative. I'm definitely going to research more about this, but just stay curious, do your research on topics, even if you don't get them and they don't click. There's a lot I don't know about this, but it was in the news, so I decided to deep dive into it myself, and still have a lot more to look into, that's for sure. But cool. Enjoy your Thanksgiving, the rest of your week, and yeah, see you tomorrow.